My name is Andy Ding. I am a laboratory genetics and genomics fellow at Mayo Clinic. Today, I am excited to talk about using nanopore long-read sequencing to resolve intragenic duplications involving the DMD gene. These are my disclosures. The first question one may ask is, why are we interested in the DMD gene? Well, DMD encodes dystrophin, which is an essential protein for muscle function. It is located on the X chromosome and is associated with a genetic condition known as Duchenne muscular dystrophy. This condition is fairly common, affecting roughly 1 in every 3,600 live male births in the United States. However, its consequence is highly devastating. These patients are usually wheelchair-bound by their teenage years and pass away before age 30. The gene DMD is also associated with a milder form muscular dystrophy known as Becker muscular dystrophy. Because genetic carriers of Duchenne and Becker muscular dystrophy are fairly common in the general population, the American College of Medical Genetics and Genomics recommends carrier screening for DMD in females who are currently pregnant or considering becoming pregnant. The second question one may ask is, why are we specifically interested in intragenic duplications? And this is because the clinical interpretation of this particular category of variants is highly challenging. You may be very familiar with this analogy of different types of variants, such as if you change a letter in a word and you change the meaning of the sentence, that's a missense variant. And if you remove several letters from the sentence and you create a deletion. And if the number of letters deleted is not a multiple of 3, this will lead to the disruption of reading frame and therefore leads to loss of function of the gene. Interpretation of a deletion is fairly straightforward because as long as you know which letters are deleted, you would know its clinical consequence. Well, not so simple for duplications because clinical interpretation of duplications is dependent on its structural configuration. A duplication, as an example, 5 letters here, can be in the tandem configuration. In other words, the inserted segment is immediately downstream of the original segment. And in this case, because 5 is not a multiple of 3, this will lead to a disruption of the reading frame and consequently loss of function of the gene. On the other hand, however, a duplication can also be non-tandem, meaning that these 5 letters were inserted somewhere else in the genome, most likely in a non-coding region. And in this case, because the reading frame is intact, we would expect usually no impact to the function of the gene. In other words, this would be a clinically benign variant. The challenge for current clinical genetic testing methods, such as microarray, gene panels, exome, and MMLPA, is that they can all be used to determine the dosage. In other words, they all know that there's a duplication there, but they cannot distinguish the configurations. The key question we seek to answer in this study is, can nanopore long-read sequencing resolve configuration for DMD intragenic duplications? In other words, distinguishing between tandem versus non-tandem. In this pilot project, we performed high-coverage whole-genome long-read nanopore sequencing for 8 samples with clinically confirmed DMD intragenic duplications. We divided the samples into 2 groups. In the 4 samples from group 1, these patients have either personal or family history of Duchenne or Becker muscular dystrophy, and therefore their duplications are expected to be in tandem and therefore pathogenic. On the other hand, in group 2, we have 4 patients with incidentally detected DMD duplications, most commonly through carrier screening. And in this group of patients, there are no prior expectation that these variants are pathogenic, in other words, being in tandem. The workflow for this pilot project is fairly straightforward. We performed whole genome nanopore sequencing, followed by alignment, and followed by manual inspection of the region of interest in the band file. We are currently developing an automated process for the inspection and structure configuration determination. And here are just the example metrics from 5 of the 8 samples. And as you can see, the average read length ranged from less than 5 kb to about more than 18 kb. And the coverage is roughly 20x or higher. In the next few slides, for the sake of time, I will present 1 case each from group 1 and group 2. This is a case from group 1, and this is a female patient tested due to a brother affected with Duchenne muscular dystrophy. Using MLPA, we detected a duplication of exons 13 to 17 of the DMD gene in this patient. And with nanopore sequencing, we were able to clarify that this duplication is indeed in tandem. The reason we are confident is because we detected split reads that span the tandem duplication junction shown in the IGV screenshots below, and also with the red horizontal line demonstrating the duplication junction. On the other hand, in this patient from group 2, she tested due to abnormal carrier screening results, and clinical testing yielded duplication of exons 52 to 57 in the DMD gene. And with nanopore sequencing, we were able to clarify that this duplication is likely not in tandem. I apologize for a busy slide, but here is a likely scenario. That's the duplicated DMD segment is roughly 1.8 megabase pair away from the DMD gene, and it is also in the middle of a 108 kilobase pair tandem intragenic duplication. The reason we concluded about this structural configuration is we detected two groups of split reads supporting the red and the blue junction shown in the IGV screenshots below. However, I would also like to emphasize that this complex structure variant can also be equally likely in this structural configuration of instead of the DMD segment inserted 1.8 megabase pair away, it can be this intragenic region C inserted in the middle of the DMD gene 2.0 megabase pair away from its original location. Nonetheless, in either scenario, this case is inconsistent with a classic tandem duplication configuration. To conclude, nanopore long-read sequencing in our situation successfully resolved structure configuration for all 8 samples in this pilot study. And intriguingly, in all 4 samples from group 1, the nanopore sequencing revealed that the duplication is indeed in tandem. This is in contrast to group 2. In all 4 samples, the duplications were all revealed to be likely not in tandem. And therefore, the inferred structural configurations are consistent with clinical history. We expect them to be in tandem for group 1, and we have no expectation for group 2. This study demonstrated the potential utility of nanopore long-read sequencing in clinical molecular diagnostics to resolve these complex structural variants and be able to clarify its clinical significance. And in terms of future directions, the first thing, as we also heard from the presenters this morning, is that we are hoping to have better automation at our process. Are we going to have a SV caller that can, without manual inspection of the band file, be able to determine the structural configurations? We're hoping to test this in additional clinical samples to see if our conclusions from this pilot holds. And we are also interested in testing this methodology in additional regions. We're interested in the DMD region because the DMD gene is one of the largest genes in the human genome, and therefore, by chance, there are more intragenic duplications involving this gene. However, the methodologies we developed from this study are equally applicable to other clinically relevant genes. With this, I would like to thank the leadership on this project. I would like to thank the wet lab bioinformatics as well as the clinical personnel who are essential for the success of this project. I would like to thank the different laboratories within the Mayo Clinic Department of Laboratory Medicine and Pathology for contributing the samples and for running the sequencing, the sequencing experiments. With this, thank you for your attention,